Welcome to the Chapter 5, Membrane Structure and Diffusion part 1, basically, of Chapter 5. So this picture is a cartoon version of the cell membrane. We've got the bilayer. You're familiar with the phospholipid heads and tails. We've got embedded proteins that you'll hear a lot more about. These look like they might be involved with adhesion. They're going to be maybe communicating to the outside of the cell. So it looks like some of these are connecting to the extracellular matrix we talked about in the other chapter. We have these little yellow blobs. This is probably cholesterol molecule embedded in the membrane. We've got these ones that are sliced open. These are going to be pores to allow things to go through the membrane that normally would be blocked in the case that they can't get through. On the inside of the cell, we see the cytoskeletal elements. In the previous chapter, we learned a little bit about the cytoskeleton. They help give the cell structure. They help anchor some proteins. They help communicate to the information to the inside. So just focusing on what might make it through the membrane. So we've got these hydrophilic head groups that are touching the water on the outside and on the inside. And then we've got the hydrophilic head groups. We've got the hydrophobic tails on the inside that are going to prevent water-like things to go through, or anything that's not hydrophobic. So what kind of things might pass through the cell membrane? Well, they have to be non-polar, they have to be uncharged, and they have to be pretty small. It kind of rules out most things from going through the membrane. The ones that come to mind right away that could make it through the membrane would be oxygen molecules and carbon dioxide molecules, and they're just going to move through with simple diffusion. They sort of snake their way through. They have no particular charges, so they just kind of make it through that interior part. How about something like a hydrogen ion that's carrying a charge? It's really small, but that's kept from passing through because it cannot make it through this hydrophobic tail area. And then water molecules, kind of small. Used to be thought that they kind of sneak through because we know that water moves in and out of cells very quickly. It turns out that water molecules need help, and I'll talk about those pretty soon. So let's talk about the movement of the membrane. So going back to this, the membranes are not static. Excuse me. The membranes are not static. They're actually quite mobile. And there were a bunch of experiments done to prove this type of stuff. One is that they could label the membrane with a fluorescent probe of sorts, and then they shine a light on it and bleach out that fluorescence. And then they want to check how long it takes for that hole that they bleached out to basically fill in. If you have a membrane that has absolutely no changes to it, it's kind of standard membrane, they got this number here. If they took the membrane and they created it so that it has a decreased length of fatty acid chains, so remember that basically these tails got shorter, then they found that the hole closes up faster. So shorter fatty acid tails means the membrane is more fluid, or it moves more. If you increase the cholesterol, it's actually less fluid at normal temperatures. If you increase the desaturation of the fatty acids, so basically you make them more saturated, remember, make them stiffer, they want to seal up, they don't have as much wiggle room. And then if you increase the protein content, it really slows things down. They don't close up that hole very quickly. So all that goes to say that the another word that they use to describe the membrane, I'm going to write it up here, is that it's called the fluid mosaic model. And that's a good way to remember the different things that are going on with the membrane. It's very fluid, things are moving around in different directions, and it's a mosaic. It's built of all these different parts. Another thing to remember is that they're fluid in the lateral sense. There are not a lot of phospholipids or proteins that flip-flop on either side. And in fact, there's some membranes that are built with a very specific phospholipid content on one side versus another to give it some special properties. So the fluid mosaic model is usually used to describe the current belief of how the membrane's made. Another experiment to show that it was fluid was, again, they took these fluorescent markers, and this time they're marking the proteins, and they made the mouse cell green, they made a human cell red, and then they treated the cells with something that makes them fuse magically here. And in the beginning, they're just half and half, and over a period of time, they found that these proteins diffuse pretty rapidly in the plane of the membrane, and they get completely mixed up. And it appears that that mixture is sped up when the temperature is increased, which makes sense. More kinetic motion, more fluidity. So let's start talking about the proteins that are embedded in these membranes. So remember, we've got our bilayer here. Outside is water and inside is basically water. So remember that the R groups have different characteristics about them chemically, and the ones on the outside are probably going to be very hydrophilic. The ones embedded in the membrane that might be poking themselves into these tails are going to be pretty hydrophobic. Additionally, they find that most of the proteins that we're going to talk about that act as facilitators to move things across have this multi-alpha helix spanning thing. So this has 7 different barrels of alpha helices, and they're called 7 spanners in the logo. So they're basically, here's the beginning of the protein, the N terminus makes alpha helix connected to a loop, another alpha helix connected to a loop, alpha helix, and so on. So you get to that last one, and there's the C terminus, so that's the end of the protein. And basically, if you create this, you can imagine looking down this way at it. It might be making a little bit of a pore that's going to allow something to pass through that normally wouldn't be passing through here. That's one role that a transmembrane protein can play. Another role that some of the transmembrane proteins play is that they help 2 cells stick together. And usually these proteins are going to have lipids attached to them, and they're called, oh, I'm sorry. This is a glycolipid. No, ignore this word. This is a glycoprotein. Glycolipids also exist. However, the picture down here is a glycoprotein. So the big brown blob is the transmembrane protein, and on the tip is probably some sugar molecules. That's where the glyco comes from. Remember glycosylation in the Golgi? Anyway, the sugars are actually part of the recognition system between cells. We're going to get to a point where in the class soon that we're going to talk about different cells that sort of talk to each other and communicate. One of the ways they do is by binding receptors that they identify on each other and then send signals into the cell. So this is an example of a protein with sugars attached to it, and they're going to facilitate the identification and binding to each other. So before we start thinking about specifically how things pass through the membrane, we have to discuss the idea of diffusion. So diffusion is the idea that if you have something in a highly concentrated form here, it wants to go to low concentration. So diffusion is the idea that molecules want to move from high concentration to low concentration. It's just energetically stable to spread out. So we're going to talk about water moving that way. So if you look at the top picture up here, we're going to think of the membrane as this gate, and that water can pass through. And water is going to try to move from high to low, so water is going to try to move through the membrane. The results of water moving you can see in 2 different ways. If we look at an animal cell, we're going to visualize it with a red blood cell, and if we want to talk about a plant cell, we're going to use these nice little plant cells down here. They respond a little differently because of their structures. First, we want to talk about the red blood cell. The easiest state to talk about is called isotonic, and that's when the water concentration and the solutes, so we're introducing one more term over here. A solute is basically things that are dissolved in the water. A solute could be an ion. A solute could be some sugar molecules, and we'll be doing experiments with these actually in the Lab 1 osmosis and diffusion. So in this case, everything's equal. There's really no movement out of the cell of water, no movement into the cell of water. In all these cases, we're going to assume that the solute cannot pass through the membrane, and it's the water that's going to move. It's always a little bit confusing. The other thing to think about is if there is a lot of solute, it's taking up the place of the water molecules. So if you have high concentration of solutes outside, it means that there's less water outside because the solute's taking up space. So in this case, the water is going to try to move out of the cell. So if you put a red blood cell in a high salt situation, you will lose water from it. I'm going to write high salt here. On the completely opposite side, if you put the red blood cell in a low salt environment, that means that there is more salt inside the cell. If there's more salt inside the cell, that means there was less room for water molecules. So the water is going to try to move in, and the cell might burst. Although these definitions are based on salts or solutes, I find that it's easier to think about where the water is, just to confuse you more. In the hypotonic environment, so it's hypotonic outside the cell, it means there is more water, or H2O, outside the cell. And I'm going to try to highlight the O's here. O's. And hypotonic has an O in it. So in a hypotonic solution outside the cell, there is more water outside the cell, so it wants to go in to go reach equilibrium. So it puffs up your red blood cell. In the hypertonic solution, so when it is hypertonic outside the cell, there is less water outside the cell. So I've got an E there, and there's an E in hypertonic. I'm going to have to talk about this again in class tomorrow, but you can listen to this a couple times on the podcast and see if you get it. If I put my red blood cell in a place where there is less water outside the cell, the water wants to move from high concentration to low, so the water will leave the cell and leave it all wrinkly and spiny like this. So just to confuse you a little bit more, the same thing's going on with plants. And that's actually not so confusing. Here it looks like the cell shrunk up, here it looks like the cell shrunk up. It turns out that plant cells actually don't like isotonic conditions like our red blood cell does. They actually like this hypotonic condition because then the cell membrane is pushing against the cell wall and making the plant nice and stiff. Think of a celery stalk that's been left out on the counter for a long time. It's kind of leaning in this direction here. It's kind of wimpy, it's kind of flappy. It's called flaccid. If you put it in, you cut off the end and you stick it in some water, water moves into the cell and makes it nice and stiff against the cell walls. And then that word is called turgid. I can't put another thing up on this. So how is the water moving through the cell? This should be the last slide, I think. Water cannot make it through the membrane. See all these fuzzy things? That's the hydrophilic head group, hydrophobic tail, hydrophilic head group. So this is a bilayer. And this is a protein structure model of something called the aquaporin. The aquaporins were discovered not that long ago, only within the past 10 or 15 years or so. And aquaporins are basically a transmembrane protein that's specialized to allow water to go through. The Nobel Prize was given out for the discovery of aquaporins by several scientists not that long ago, maybe about 5 or 6 years ago. Here's a cartoon version of how they think it works. It's embedded in the cell membrane. They think that there are these alpha helices. We're kind of